Today, we're going to learn how to make a smear from a solid and a liquid, and we're going to do 3 different simple stains. First, we're going to start off labeling our slides. Our first slide is going to have a mixture of Micrococcus luteus, so we're going to label it M. luteus, and Corynebacterium xerosis, so C. xerosis. And this is a simple stain. We're going to be doing this with methylene blue. We'll go ahead and make this slide first. Both of these are from solid media. So whenever you have bacteria on a solid plate like this, if you think about it, bacteria are microscopic, but on this plate you can actually see the organisms. The reason you can see them is because there's so many of them. The smallest speck of bacteria that your eye can see is 1 million bacteria. So if you were to take a big chunk of this and put it directly on your slide, you would just have millions of bacteria on your slide, and it would be useless because you would actually have too many bacteria on your slide to make viewing possible. So anytime you take from a solid, first you have to dilute it. And for that reason, we have sterile water. So in order to do this, I'm going to go ahead and take my loupe and I'm going to flame it. I will insert the loupe until it's red hot. Retract it and allow it to cool. Now I will take my sterile water and, using my sterile loupe, take a drop of water and put it on the slide. You may need more than 1 drop. So if you do need 1 drop, you'll flame your loupe again and then repeat the process. I'm going to go ahead and flame the loupe since I've used it. I'm going to insert my loupe, leave it until it's red hot, and then retract it and allow it to cool. Because I'm putting 2 samples on this slide, I'm going to put 1 drop of liquid down at one end of the slide and the other drop of liquid down at the other end of the slide. With my sterile loupe, I will take my 2nd drop of distilled water, sterile distilled water, and place it down at the other end of the slide. I'll recap my bottle, and when I'm done, I will flame my loupe. I'll insert the loupe, wait for it to get red hot, and then let it cool. Now that I have my water on the slide, I can use it to dilute my bacteria. When you're taking from a solid, you want to make sure you use a very small amount of bacteria. Using a loupe has much too big of a surface area. If you used a loupe, you would take a ton of bacteria, and that would be too much. So what we're going to use is a needle. So I'm going to go ahead and flame my needle. I will take my needle, insert it until it is red hot, and then remove it and let it cool. Here I have my plate of Micrococcus luteus. I'm going to take my plate and open it And I'm going to take the needle and just touch it to one of the colonies. Notice I'm not making a big gouge, I'm just gently touching it. And you shouldn't even see any bacteria on the needle. I'm going to take that and now mix it into the water on my slide. And I'm going to use my needle to break up any clumps that there might be and to spread that liquid out. I want to spread it out because the next step is to air dry it. And the thinner you have it in this step, the faster it's going to air dry. Now that I've used my loop, or my needle, I'm going to go ahead and flame it so I can get a sample of my next bacterium. Going to take my needle, insert it into the back of the incinerator, wait for it to get red hot, and then I'm going to go ahead and retract it. Now I will put my second sample of bacteria on the slide. Here I have Corynebacterium xerosis. I'm going to take my plate, touch my needle to a colony, and mix that into the water down at the other end of the slide. I'm going to break up any clumps, spread it out thinly, and set it aside to air dry. And after using the needle, I'm going to go ahead and flame it. As always, I'm going to insert the needle, allow it to get red hot, and withdraw it and let it cool. While my first slide is air drying, I'm going to go ahead and prepare my second smear. On this smear, I'm going to be using Staphylococcus aureus, and Bacillus megaterium, and this is going to be a simple stain, and I'm going to be using safranin. My Staphylococcus aureus is from a solid, so I'll have to add a drop of liquid. My loop is already sterile because I've flamed it before, so I can go ahead and use that now. I'm going to use my loop to take a drop of my sterile water, and I'm going to put the sterile water down at one end of the slide. And after I'm done using that loop, I'm going to flame it. Insert the loop until it gets red hot. Withdraw and allow to cool. My needle is already sterilized because I flamed it before, so I can go ahead and use the needle now. I have my sample of Staphylococcus aureus. I'm going to go ahead and take my sample, just touch it to a colony, and mix it into the water on the slide. I'm going to use my needle to spread out the bacteria. And because I used my needle, now I'm going to flame it. I will insert until it's red hot. Withdraw and let cool. Now the other bacteria that's going to be on this slide is in a liquid form. When bacteria are in a liquid, you don't have to add water to dilute them because they're already diluted. So for this, I'm just going to take my loop to put bacteria directly on the slide. And it's already sterile because I flamed it after I used it. Here's my tube of Bacillus megaterium. I'm going to go ahead and mix it either with a vortexer or with my finger. And I'm going to take my sterile loop and aseptically put a loop full of bacteria on the slide. I'm going to use the loop and spread that bacteria out thinly so it will dry. And I will set that aside to dry. Now that I've used my loop, you know what I have to do. I'm going to flame my loop, let it get red hot, and there we go. While that slide is drying, I'm going to go ahead and prepare my third slide. This slide is going to have E. coli and Enterococcus faecalis. It's going to be a simple stain, and it's going to be done with crystal violet. Both of these are in liquid, so I won't need to use the distilled sterile water at all. They're already diluted. So I'll take my loop that I already had sterilized and use that to get my first sample. So my first sample is E. coli. I'm going to go ahead and mix it with my finger or a vortex and take my sample. I'm going to go ahead and put this down at one end of my slide and use my loop to spread that bacteria out. Again, the thinner I spread it now, the faster it will air dry. And of course, I have to flame my loop. I'll put it in the back of the incinerator. Let it get nice and hot. And then retract it. A loop to take my second sample of Enterococcus faecalis. Mix it up either with a vortex or with my fingers. Take my second sample. And use my loop to apply the bacteria to the slide. I'm going to spread it out so that it will air dry. And now I will do one final flaming of the loop. Now that we have the bacteria on our slides and we've made our smear, the next thing we'll want to do is heat fix the bacteria. What that means is applying heat to the slide so that the bacteria are killed and so they stick to the slide. If you think about baking, if you're baking a cake or something, you always want to grease the pan before you bake it, otherwise the food will stick. Here, we're going to essentially bake the bacteria to the slide without greasing the pan. So this is how you heat fix. I'm going to take one of my slides with the bacteria facing away from the bacteria incinerator and just put it up against the surface for 5 to 10 seconds. And that's sufficient to fix the bacteria to the slide. So I'll go ahead and do it with my next slide. And then I'll do it with my final slide. And now those slides are ready to be stained. Here I have a staining tray, and I'm going to go ahead and put my slides on the staining tray. This slide is methylene blue, so I'm going to take my methylene blue and just coat it where I have the bacteria. And I'm going to set a timer for 1 minute. My next slide, the middle slide, is Frannan. So I'm going to go ahead and use this. This is a pink color. So I'm going to go ahead and use this. And again, just cover the slide with the stain. And that also will be 1 minute. And the final slide is Crystal Violet, which is a purple color. And I'm going to go ahead and cover that and allow that to sit for 1 minute. Now that it's been a minute, I'm going to take my slide and with this water, gently rinse it off. Notice I'm not fire hosing it. I'm letting it rinse off. I'll do the same thing with my second slide. Just allow the water to rinse down. And I'll do the same thing with my third slide. The final step is to blot them dry with bibulous paper. This is bibulous paper, and to use it, you take a slide and insert it and gently press. You don't want to press too firmly or you'll break your slide. We'll do it with the second slide. Just gently blot, and that removes the water. And then we'll do it with our third slide. And now we're ready to look at them under the microscope. You'll see those results posted for you online. That's how you make a simple stain and smears from solid and liquid. Take care, and I'll see you next time.